Across the planet, species distributions are shifting in response to climate change. Shifting species ranges can lead to negative and unpredictable consequences, such as the loss of biodiversity, ecosystem services, and the introduction of diseases into novel environments. It is therefore of urgent importance that we can accurately predict and plan for distribution shifts. Early predictions of climate-related range shifts assumed that species would track warming temperatures by migrating towards the poles. However, plants and animals around the world have shown a wide variation in response of their distributions to changing climates. The absence of uniform distribution shifts indicates there are other factors interacting with climate change, causing the variety of modified ranges. In the past, it was often assumed that either the abiotic, the non-living aspects of the environment, or biotic interactions like competition and predation could limit and change species distributions. However, these predictions do not generate the complex range responses we've observed. To address this shortcoming, my research instead asks how the interactions between abiotic and biotic factors affect range changes. Hi, my name is Christian Brown, and I'm a PhD student at UGA's Odom School of Ecology. In my previous work in the Appalachian Mountains, I saw how interactions between climate and biotic factors could affect species distributions, which inspired me to investigate the mechanisms behind these shifts and what their effects might be at larger geographical scales. In terrestrial environments, tree species distribution shifts are particularly important as they often serve as foundational species in their ecosystems by determining community structure and stabilizing ecosystem processes. When lost, tree species can have cascading negative effects throughout an ecosystem. My research investigates the interactive role biotic and abiotic factors play in determining the range shifts of tree species in the eastern U.S. by using a combination of experimental and quantitative approaches. In a large-scale experiment, I'm planting tree seedlings at different levels of biotic and abiotic stress from Whitehall Forest in Georgia to Harvard Forest in Massachusetts to see how these seedlings respond to interacting stressors when they are within their native range versus beyond it. To complement and expand the scale of the experiment, I'm using cutting-edge computational models across 80 tree species to understand how the importance of biotic versus abiotic interactions shifts as you move from one edge of a species distribution to another. Understanding these range change mechanisms is crucial for accurately predicting when and where species distributions will shift in the future. Conservation, land, and sea managers, as well as disease control agencies, are all currently making use of future distribution predictions to plan and prepare for negative outcomes resulting from range changes. My research aims to improve our ability to predict distribution shifts and thereby our ability to conserve and protect our environment in this time of rapid global change. Thank you for your consideration for the ARCS Scholarship.